My name's Chris, and we're going to dive deep into the world of cellular biology and information science to see if we might get a little closer to the big question, where did we come from? New York City is a pretty busy place. The traffic, the people, the messaging, the systems of transportation. But when comparing a living cell to this great city, New York pales in comparison. A cell is so complex that scientists have only scratched the surface of what really goes on in there. However, even with our limited understanding, what we do know is that the inner workings of a simple cell are simply extraordinary. One particular protein, called a motor protein, transports various cellular cargo, like energy-producing mitochondria to cellular neighborhoods in need of fuel. They can also provide the pulling power needed to separate chromosomes during cell division. Amino acids carry information from one part of a cell to another, as well as to other cells within the organism. So as you can see, without amino acids and proteins, basic cell structure as we know it could not exist. Other important types of proteins are enzymes. Enzymes are catalytic proteins that have special slots which hold other molecules to make chemical reactions possible. There are over 2,000 enzymes, and each one is used to enable a chemical reaction without ultimately being altered itself. It's been observed that the slowest biological reaction would take 1 trillion years without an enzyme, but the same reaction takes only 1/100th of a second when the enzyme is present. Pretty impressive. Adenosine triphosphate, otherwise referred to as ATP, is the energy source the enzyme uses to enable the reaction. Living organisms require and manufacture these enzymes along with all the other proteins. Let's briefly examine how proteins are created.
This animation demonstrates how the digital information encoded within DNA is used to direct protein synthesis. This is a DNA double helix containing the digital code which directs the cell in all aspects of operation. And here we see a protein complex called an RNA polymerase traveling down the DNA strand. As it moves down the strand, it carefully unwinds the DNA, preparing it for transcription. Inside the polymerase, we see a single-stranded copy of the original instructions being assembled as individual bases are positioned and added to the growing strand. A stop code marks the end of the protein specification, at which point this copy, known as a messenger RNA transcript, exits the polymerase and heads towards a two-part chemical manufacturing machine called the ribosome. While the messenger RNA moves towards the ribosome, transfer RNA molecules attach to specific amino acids in preparation for assembly. As the messenger RNA transcript passes through the ribosome, the process of translation begins. Using the instructions encoded on the messenger RNA as a template, the transfer RNA molecules align specific sequences of bases to corresponding amino acids, creating a protein chain. As this chain exits the ribosome, it is met by chaperones which prevent premature folding, while escorting the protein to a barrel-shaped machine called a chaperonin. This machine helps fold the protein into the precise shape required to perform its function. Although it is unclear how the chaperonin achieves this, we do know that accurate folding is essential in order for the protein to accomplish its intended function. Once the protein is complete, it is released into the cytoplasm to do its job.
As you can see, protein synthesis is an amazing process which occurs constantly in our bodies. Now, get this. Ribosomes are made from RNA and proteins, so the ribosomes are made of the very proteins they manufacture. In fact, it takes over 150 existing proteins to manufacture just one protein. So in essence, it takes a protein to make a protein. It's this chicken and egg scenario that continues to baffle scientists.
There are thousands or millions of interacting computers in every cell that communicate with one another. They read the information, they transfer information from one component to another component. There are many different operating systems involved. There are different programming languages involved with the various components that are within the cell. And all of them have the same components that any other computer would have.
The necessary and sufficient components of any functional computer are memory for data storage, an executable program containing instructions for processing data, the processor, which executes the instructions, and the capability to produce meaningful output. Many components of cells are components of real biological computers, equivalent to the components of electronic computers. For instance, DNA and RNA can hold prescriptive information or algorithms, and proteins can be used to hold, transfer, or process data. Proteins are the output generated by the translation process of a ribosome's computer system. Scientists have also discovered something known as the interactome, which is an incredibly dynamic network used by the cell for internal communication. It's like the internet of the cell, where proteins, RNA, and DNA can stay in constant communication with each other. We also know there is a cell-to-cell exchange of information integrating our entire body. When cells were discovered over 300 years ago, it was thought that the structure of a cell was very simple. With the inventions of the electron and proton microscopes, we now have a much clearer understanding of the intricacies of life. The cell is an extremely efficient and well-networked computer system with thousands or even millions of processors and billions of bits of information. It can take in nutrients, convert the nutrients into energy, carry out specific functions, and reproduce itself as necessary. Even more amazing is that each cell stores its own set of instructions for carrying out each of these activities. Let's take a closer look at how a cell works.
The first thing we see is the cell membrane. Much like the firewall software on your computer, the membrane contains protein gatekeepers, allowing only those components into the cell that belong and rejects all other components. Once we pass through this membrane, we see the organelles, or the organs of the cell. Ribosomes are the computer-controlled protein manufacturers of the cell. By fastening to an mRNA and using it as a template, the ribosome arranges amino acids in the correct sequence to form a particular protein. This process is known as translation. Golgi body. Next, we find a Golgi apparatus, also known as a Golgi body. Found in both plant and animal cells, Golgi bodies package and store proteins. Much like a post office, the Golgi packages and labels items, which it then sends to different parts of the cell. It's composed of membrane-bound stacks known as cisternae. Each cisterna is made up of a flattened membrane disc, and its primary job is to modify proteins, but it's also involved in the transport of lipids around the cell and the creation of lysosomes. Lysosomes. Lysosomes are spherical organelles which dispose of all the waste in the cell. These tiny organelles contain acid hydrolase enzymes that break up waste materials and cellular debris. The membrane around the lysosome allows the digestive enzymes to work at the 4.5 pH they require to break down the debris and dispose of it. Without the lysosomes, the cell would become overwhelmed with debris and ultimately self-destruct. Mitochondria. Mitochondria are the power plants of the cell, generating most of the cellular energy known as adenosine triphosphate, or ATP. In addition to supplying ATP, mitochondria are involved in a range of other processes, such as signaling, cellular differentiation, cell death, the control of the cell cycle, and cell growth. Nucleus. The nucleus is the control center of a cell. This is where we find most of the DNA which contains the instructions needed to create other elements of the cells. Like the motherboard of a computer controlling its components, the nucleus directs the growth, metabolism, and reproduction of the cell, among other things.
Let's examine what DNA is and how it works to better understand how information is used in the cell. Deoxyribonucleic acid, or DNA, is a long polymer of nucleotides, each nucleotide being a deoxyribose sugar molecule, one phosphate group, and one base. Nucleotides are joined together to form the DNA backbone. A second DNA strand then joins the first to form a double helix. You may recognize the code letters A, C, G, and T, which are the 4 bases used in DNA. These bases are adenine, A, cytosine, C, guanine, G, and thymine, T. Like the code in software, the sequence of the 4 bases encodes genetic instructions used in the development and functioning of all known living organisms. Once organized, this information is what makes up the gene. And genes, as we all know, contain the recipes for the components of life. Genes hold all the information to build and maintain an organism's cells, and they also pass genetic traits to offspring. Human DNA contains an estimated 20 to 25,000 genes in its approximately 3 billion base pairs. And did you know that if the DNA in an adult human's body were laid out end to end, it would be about 30 billion miles long? That's enough to travel the distance from the Earth to the Sun over 320 times. A gene contains both coding sequences that determine what the gene does and non-coding sequences that determine when the gene is active, as well as thousands of other regulatory functions. When a gene is active, the coding sequence is copied in a process called transcription, producing a messenger RNA or mRNA copy of the gene's information. mRNA is produced by a protein that's over 3,000 amino acids long as it reads the genetic code. Wait a minute. How can messenger RNA require a protein that reads the genetic code, but yet the protein needs the messenger RNA to be produced? Could these have both come into existence at the exact same time with the meaningful information needed for both to operate properly? As you can see, much is still a mystery when it comes to the origin of this complex organic language.
And what's fascinating to me, not only do proteins create proteins, but we have proteins that are actually creating the DNA molecule. That's our blueprint. And yet on the DNA molecule, there are sequences of part of the DNA that are coded for those proteins. So the manufacturing plant needs the blueprint, and the blueprint, the DNA, needs the manufacturing plant in order to actually create the DNA. So what came first, the proteins that made the DNA or the DNA that made the proteins?
Once DNA is organized into a single coiled piece, it's then wrapped around a complex hierarchical structure of proteins. This is what is commonly known as a chromosome. There are 23 pairs of chromosomes in the human genome. Living organisms continually produce new cells through a process called cell replication throughout their entire lives. And here's how it works. First, we start with our DNA double helix. Helicases are molecular motors that use the chemical energy of ATP to break hydrogen bonds between bases and unwind the DNA double helix into single strands. This process is used for both replication and repair of the strands. If unwound, a single DNA strand would be about 6 feet in length. And yet, it fits into a cell which can't even be seen with the human eye. Remarkably, these strands are kept from tangling as they are separated and duplicated. Next, these strands are copied by enzyme-based computers, creating an exact replica of each original strand. Over 30 different proteins are required for duplication and repackaging of cellular components during cell division. It's from this tiny, masterfully complex cell that life is formed. While we know a great deal about the cell and its functionality, we've only scratched the surface of its inner workings. Scientists are continually discovering new information and data concerning the cell and its DNA. As our knowledge increases, it gives us a deeper understanding of how complex a single cell really is. And as we ponder this miniature marvel, we begin to wonder, could this simple cell have evolved from the elements of the earth over time? Is it possible for this to have happened by natural processes? So, what is the probability of a simple cell evolving by undirected natural processes? The probability of a single protein being formed by undirected natural processes is only 1 in 10 to the 164th power. That's 10 with 164 zeros following it. That's pretty big. The probability of life, a simple cell evolving by undirected natural processes, is 1 in 10 to the 340 millionth power. That is unimaginable. Here's an illustration to help demonstrate just what this means. Here we have one tiny grain of sand. It's been estimated that there are 1 million grains of sand in a half a cup. It would take 1 million half cups of sand to fill a swimming pool that's 6 feet deep and 30 feet in diameter. Now, if we took 1 billion of these pools of sand, we could fill Lake Tahoe in Nevada, which is about 22 miles long and 12 miles wide. Think you could find that original grain of sand we started with? We're not done yet. It would take about 1 billion Lake Tahoes to fill the volume of the Earth with sand. The probability of grabbing that original grain of sand out of the Earth filled with sand is 1 in 10 to the 30th power. It would take 100 million Earths to fill 1 sun with sand, and 1 trillion suns to fill our solar system, 10 trillion solar systems to fill 1 cubic light year, 100 trillion cubic light years to fill the volume of the Milky Way galaxy, and finally 10 billion Milky Way galaxies to fill the observable universe. Whew. That is a lot of sand. The probability of now randomly picking our original grain of sand from the entire observable universe is 1 in 10 to the 96th power. Still a far cry from our protein being formed at 1 in 10 to the 164th power, and even less chance of life evolving from undirected natural processes at the probability of 1 in 10 to the 340 millionth power. Starting to get the idea? Scientists generally consider anything with a probability of less than 1 part in 10 to the 70th power operationally impossible. And by the way, that calculation doesn't take into consideration the information that's stored in the cell, which directs the cell in all aspects of operation. That estimate is solely the chance of chemicals combining to form something living. We look at the ideas of the Earth being flat, or being the center of the universe, or the cell being the simplest component of life. And while these theories seemed promising at the time, we have discovered they are completely incorrect. As we learn more about this amazing world in which we live, we start to understand the complexity of its workings. And in the case of the cell, the more we research, the more complex it seems to be. As we gather information, it is up to us as scientists, students, and colleagues to bring science to a level of integrity and critical examination that it deserves. If we approach science with an unsupported prearranged bias, then what we're trying to accomplish is not really science at all. The beauty of science is that we're able to move away from accepted dogma to examine the evidences. It's not up to us to disprove a given theory. It's up to the theory to prove itself against the laws of science. If a theory fails to do this, then it should be rejected, and we should search for more knowledge in order to, as Sigmund Freud said, discover the entire truth. The possibility of life evolving using the known laws of chemistry and physics is operationally impossible. When we consider the laws of information, that possibility now becomes impossible. Meaningful prescriptive information cannot arise from nothing, no matter how much time you allow. And until we acknowledge this, we will never discover the origin of life. Thank you for joining me on this amazing journey through science and the programming of life. I would challenge you to examine the evidences and join the growing community of critical thinkers. Until next time.